slew of different yeast strains that I kept in the -80 freezer. And then every month when we had our club meeting, I would make, a dozen plates of all different varieties of yeast and bring them in and distribute them to the different members and give them instructions on how to make their own starters. So back then I was brewing all the time, but now, my batches are, one here, one there. And then now that it's cider season, I'm making hard cider every once in a while. It's so easy to make the hard cider. So
yeah, no, I was curious because you're talking about quick beers, and the Kvike yeast is extremely fast. That farmhouse style. I recently started playing with it, and I did this APA I'm drinking right now. I think start to finish, it was ready to go in 5 days. I actually felt bad. I gave it a couple extra days. It was probably ready in about 4, and it just came out fantastic. And that stuff's a beast.
What style of beer is it?
This is an APA. So not an IPA, but an APA. And it actually, this is not filtered. Oh, there we go. Zoom is playing with my my background there. That's actually my brewery in the background, but I'm actually sitting in my office.
You still got pretty good clarity on it though, even though it's not filtered. It came out pretty clean.
So what was your hop build in that?
In this one, there was actually, I used a handful. There's Centennial, a little bit, I'll have to pull up my recipe. I actually made it a couple of months ago. I brew at the half barrel scale, but my wife stopped drinking beer around the same time. So I've been drinking the same APA now for over 2 months. I
see. Yeah, so when you start getting these hoppy beers, what I found, especially with the New England style IPAs, which have so much aroma in them, is it's got the shelf life of dairy products. That's why it's so important when you go to buy one of these at the store. I have to bring my reading glasses so you can look at the bottom and find out when they canned it. Because if it's like 3 or 4 weeks old, it's just not even worth it to buy because there's no hops left.
Yeah, it has a very short half-life on those hop bombs. Yeah.
I feel like this might be the right place to ask this. So a lot of my local breweries, I'm in the San Francisco area, have started canning their own crowlers and stuff at the tap. Can anyone give me some guidance on the shelf life of something like that compared to a traditional canning operation? Is it also quite short?
So it all depends on your style of beer, right? So hops and oxygen do not go together, and it's really, really hard to get beer from one container to another without introducing just even the littlest bit of oxygen. If there's any oxygen there, your oxidation is going to kick in and your hops are gone, especially the aroma. The aroma will be the first thing that disappears, and then the taste will start to go. The bitterness is just going to stay there, right? But especially these Northeast New England, the NIPAs, but IPAs in general. As soon as you do that transfer, unless they have one of those systems where they purge the whole container with carbon dioxide first, and then do the decant where it goes in slowly, displacing the carbon dioxide, you really want to get to it in a day or 2.
Cool. No, that's great to know. Thanks. I don't think, a lot of the ones around here are not that sophisticated. They've just established kind of haphazard canning operations in the last 7 months, obviously. So
if you go in, if they...
John, can I just jump in here? Yeah. I want to make sure we do introductions first, and let's continue where we pick up because I feel like we're getting so excited jumping in. So hold that question. So real quick, I want to kick this off. So welcome everybody to the SLAS Science of Beer Making Meetup. As you can see, we're kind of already like right into it, which is super cool. I'm loving that everybody has an excitement. We're going to start asking a ton of questions. So I'll keep this short so we can get to talking about the beer. Really, this is just kind of a casual format. Again, John, thank you for your time and coming in. You're just, already spouting so much wisdom, which we're looking to soak up. So real quick, my name's Coleman. I'll be moderating and looking out for questions. I've been a part of SLAS since 2013. I'm part of a startup called Farologics, but the reason I'm here is because I got into home brewing about 5 or 6 years ago. So I'm still relatively new and learning a lot. I love IPAs and Belgian ales. But with that, John, why don't you give us a little bit of your background, both kind of professionally and how you got started with home brewing?
Sure. So I did my PhD from, I don't know, I finished up at the University of Rochester with PhD and then a 2-year postdoc with a PhD in yeast molecular biology. So I was growing yeast. And during my graduate program there, I met some people who were brewing beer, and they were part of the Upstate New York Homebrewers Association. And when they found out that I did yeast molecular biology, my field of study for my PhD was how yeast replicate their DNA. And so I characterized one of the DNA polymerases that's involved in the initiation of DNA replication. And then for my postdoc, I worked on a DNA polymerase that that deals with repair of damage to your DNA, like when you sit out in the sun or get exposed to X-rays or things like that. And so for all of these experiments, yeast was just a model system because they've got all the same enzymes that we have, and it's easy to do all kinds of manipulations of the yeast. But in addition to that, I've got all my yeast strains. I store them all in the -80 freezer forever, and you can make plates and grow up cultures and purify proteins. And so when they found out I could do that, the local homebrewing club, which had like about 100 members, it was a pretty, it is still a big club in Rochester, New York. They asked me if I would join the club and become the librarian for a yeast library. So I did. We joined the club, and I started, and I thought about making beer because it didn't seem like it could be that much more complicated than growing 14-liter batches of yeast for purifying proteins. And it turned out it wasn't. And so I then established and this was way back in 1987, right? 85, let's see, started in, I think 87. And so the homebrewing market back then was really, really difficult. You had a hard time finding high quality products. You could find some dry malts out there, but batches were kind of flaky, and you could find some of the syrup malt extracts out there. But it was really hit or miss whether you get exactly what you wanted. And you really couldn't store things for any length of time. Things would grow in the stuff you purchased. And so there was a lot of word of mouth, and this was really before the internet was a big thing. So you couldn't just Google something and find it. So there was a lot of information back then that was just sort of passed on by word of mouth. So I joined the Homebrewers Club. I started teaching all of the members there how to make their own starters once I figured out what the right chemistry was, what the right series of events was to get a good brew started off and get it done in the right period of time and have the yeast be as happy as possible. And I read a ton of books, and what happened was, as soon as I started supplying all of the people in the club with plates and instructions on how to make the right starters, everyone started winning competitions all over the place. And so it really took all of their beers from something that was drinkable to things that were winning competitions out there. So that was back then. I've been brewing ever since. I've consulted for a couple of breweries along the line. Usually I teach them how to use a microscope and how to tell the difference between what a bacteria looks like and what a yeast looks like, and what's a flocculating yeast strain versus a non-flocculating yeast strain, and how to do counts with a hemocytometer so you know what numbers you've got. So that's me. So I've been brewing beer for 30 years or something.
Man, that just sounds like a blast, and you've had such great experiences. I mean, it seems such like so much fun. Before we get too into the details, and I want to get back to Nick's question about oxidation, because I think it's a big factor, particularly in the beer production process. But could you just give everybody like a general overview of how beer is produced and some of the key challenges that go into homebrew production?
So I'm assuming everybody knows that yeast do not eat starch.
We'll go from there. Yeah.
So beer is made from barley. Barley seeds are full of starch. And so there is a chemical reaction that has to happen in order to make sugar from the starch. And the sugar is what the what the yeast eat to turn that into carbon dioxide and ethanol. And so you get barley seeds, and their malted barley is a seed that has been germinated, so that down at the base of it, a new plant wants to start growing. And that new plant, just like yeast, needs sugar, not starch, but the seeds are all made of starch. And so they take the seed that's ready to grow, they put a little water on it, they let it germinate, and then they quickly dehydrate that seed, and they call it malted barley, and that's what they sell to brewers. Now, a brewer gets a seed that has two different things in it. It's got all starch at one end, and it's got all these enzymes at the other end that are ready to turn the starch into sugar for the plant to grow, but they've killed the plant by cooking it. So now you grind that grain up to kind of powderize the starch, and then you make oatmeal from it, kind of a warm mash. You add water to it to warm it up. And all of the amylases, the enzymes in there that eat starch and turn it into sugar, now become active. And so this is your mash, where you're allowing the enzymatic reaction of turning the starch into sugar to happen. And when that's done now, you've converted some proportion of the starch into sugar. There's usually still a little bit of starch left over. And that is what you're then going to use to boil for a while. And then after it's done boiling, hey, Chris, you're going to add in your yeast to then eat the sugar and turn it into alcohol. So if you're a home brewer, you can buy a liquid extract, which they've already done the germination, they've already ground up the stuff, they've already done the mash, they've already turned the starch into sugar, and then they suck all the water out of it and make it into a really thick syrup that you can buy in a can or a bag or something like that. And so for home brewers, that's a really convenient starting point, because now all you got to do is add water to that sugar mixture, boil it with your hops, and then let it cool down and throw in your hops, and a couple days later you got beer. So for home, that's when I started home brewing. That was what I did. Was I would buy the liquid extract, I would add water to it, heat it up, throw in my hops at different times, let it cool down again, throw in some yeast, and go away for a week, come back and put it in bottles. Wait another week, and then you got beer. And as a graduate, as a broke graduate student, that was a great way to get beer.
Excellent. Nick, I know I cut you short there, man. Did you want to jump back in with your question on oxidation?
Yeah, it was mainly the comments earlier on shelf life and freshness of beer. And I don't know if you guys are the same, but certainly trying to support local breweries recently, and just wondering what shelf life looks like from something I pick up down the street versus something canned in a more rigorous commercial environment. But I think John answered the question mostly. I don't know if he had anything else to add. But
so it's important to know that oxygen is your friend when you start making your beer, and then it's your enemy once you've made your beer. So yeast are so when you're trying to make beer, the yeast need oxygen at the very start. You've got that starter that you use for starting off your brewing process has really got to go through one or two doubles in that wort that you've made in order for the yeast to get happy, for them to make their equivalent of fat, and it takes oxygen for that to happen. And so after you've done this 1 hour boil of your wort to get the sugars converted and to get all the acid out of your hops and to get the flavor out of your hops and to get the aroma out of your hops, you're then going to drop the temperature down. And then before you throw your yeast in, you want to you've got to aerate that and get a ton of oxygen in there, because now you're going to throw your starter in, and you want those yeast to grow aerobically. You want them to be using oxygen to help them convert that sugar that's in there into, you know, a couple more yeast. You normally want your yeast to double, like 1 or 2 times is what I say is the minimum. You don't want them to only double once, but they have a maximum density that they want to get to in that wort as they're fermenting. And the more yeast you put in, the fewer yeast they have to make to get to that maximum density. And one of the things that kills your beer, that makes it taste, that gives you all these weird diacetyls and weird chemicals that are considered off flavors in beers, is if you put in a couple of yeast, not very many, and they double, and then they hit maximum density, and then they just sit there and convert sugar into alcohol. All of those doubles, every time they go through one of those doublings, they're making byproducts that don't taste good. So the perfect starter for a batch of beer, for most beers, is about 2 or 3 doubles before maximum density. So what I use when I'm trying to make a beer for a competition is I'll do 2 batches of beer. I'll do my beer that makes my yeast, and that'll be like an okay beer. But then I'll take a quarter, maybe an eighth of the trub from that beer, and the trub is the yeast. So after you've dumped your yeast into the first beer, you've let it ferment completely, the fermentation has stopped, you've got this inch of solid yeast packed down at the bottom of your beer. You take your beer, you transfer it to a new container, and then that gets, maybe it ages for a week, and then you put it into a keg. But that yeast from the bottom, that's your trub, and that to me is pure gold, because that's a big fat chunk of really happy yeast that just grew in the last 2 years, and I'll take a quarter of that and use that as my starter for my next beer. And I know that it's a quarter, so that means it can double, and it can double again, and then it's at maximum density in that new beer. And that's only happened in, like, 3 hours. And now it just sits there and converts sugar into alcohol. So the way you make a good beer is not by making a whole crap load of yeast. It's by putting a whole crap load of yeast into a whole crap load of sugar and making beer. And so that is one of the quality control things that smaller breweries have a difficult time with, because it's difficult to get that chunk of yeast to use as your starter that starts you out right at the correct density. And so, my rule of thumb is that yeast are going to get to something like 2 times 10 to the 8th yeast per mil. And so I'll pitch in 2 times 10 to the 7th, and that will double to 4 times 10 to the 7th, and to 8 times 10 to the 7th, and maybe it'll get to 2 times 10 to the 8th, but that's it. And then that'll just sit there and process sugar.
Awesome. All right. Any other questions you guys want to ask? Oh, floor is open. All right. I got a question, John. So in your career as a scientist, and are there any specific ways that's like influenced your practices at home? Did the aseptic technique you learned in grad school transfer to making beer, or how much overlap have you seen?
It's really funny. So in the lab, I'm really careful with sterile technique, but when I'm making beer, not so much, because you're dealing with like an army, right? So I never sterilize anything at home. I don't sterilize my carboys. I don't sterilize my body. They just all get rinsed out with hot water. I don't let mold grow in anything. Now, if I've got a mold growing in something, that's a whole different story. But when I'm done drinking a beer out of a bottle, if I'm going to bottle beer, I usually don't bottle. I usually keg now. But occasionally I want to pull off a bottle for a friend, and so Nick, I will fill up a bottle from a tap, I'll cap it and I'll bring it over and tell my buddy, you got to drink this today. Because there's oxygen in there and it's gonna oxidize the hops. But I clean out everything with hot water. And the thing you need to know is that beer, if you use a good starter, you're putting in just a gazillion yeast, right? And they're going to take that sugar water and they're going to immediately drop the pH. They're going to raise up the CO2 levels in it, and there's going to be alcohol there. And that is the triple combination that kills all bacteria, pretty much. There's nothing lethal that can grow in that combination, which is why beer judges know that while what they're sipping might taste like crap, it won't kill them.
Well, and the alpha acids, I'm sure, from the hops also contribute, right? A little bit.
Yeah, to some extent. There's, that's acid, and the yeast are dropping the pH down to somewhere around 4, just because of the fermentation process. And so, but yes, certainly the reason why IPAs were invented was to make sure that and that I think helps a little bit with mold. Mold will grow in things like that, and so having a little bit extra to help prevent mold, to transfer those barrels over to India from England back in the day, didn't hurt.
So what types of species? Go ahead. Keep talking.
So I was going to go back to oxidation one more time. I'll tell a story. So my wife and I went to there's beer festival when there's not COVID pandemics going around. There's beer festivals all the time. We went to a beer festival one time in central New York, and it was like a 95-degree day. It was a beautiful day to walk around from one tent to another drinking beer. And we went to this one booth where, and I don't know what the brewer wouldn't tell me what strain of hops was in his beer. And this was, I don't know, probably in 93 or something like that. It's a long time ago. But so, you get a baseball cap when you come in, and if you're wearing the baseball cap, you've paid your entry fee, and they give you little tasters. And so, we got a little taster cup, and I tasted it, and I was like, oh, it's a pretty good beer. It was a German style. I think it was like a Kolsch style beer. And then I walked over to the next tent over, which was 15 feet away, took a sip from it. It was completely skunked. 30 seconds. So I walked back to the guy and I was like, dude, you've got a hop that's really sensitive to sun, and there's not a cloud in the sky. He's like, what are you talking about? And I said, tell you what, do the experiment right here. Pour 2 tasters. So he poured 2 tasters, and I took them and I set them down on the table, one in the sun, one not in the sun. And I had him count to 30. And then I took them both out and I said, okay, taste the first one. He tasted it. He's like, yeah, that's my beer. I'm like, taste the second one. He's like, holy shit. It was so bad. It was so bad. And so from that time on, we started telling all the people there about this, and everybody was walking around with their baseball caps over their beer instead of on their head. So there's certain hops that are really, really sensitive to, and they oxidize just immediately as soon as they get hit by sunlight. It must absorb the UV light in the sun. But so that's a thing. And so I always tell people, and now everybody's drinking beers outdoors because everybody's drinking at these outdoor tables at the bars. And so I always remember to bring my mask and something to cover my beer when I'm sitting outside drinking beers at the outside tables.
So what I'm hearing is that we need little baseball caps for all of our beers.
Yeah, I think you've stumbled on a new product here. Maybe we could do SLAS like beer sun umbrellas.
Well,
something like that.
That cuts towards why commercial beers are typically, not all, but typically distributed in brown or green bottles. Very few you'll find in clear bottles, and the ones that are in clear bottles typically are the skunkiest beers. And there's definitely a connection there. There's a photochemical reaction that starts, just like you said. And so if it's in a light blocking container and aluminum cans, you see a lot of commercial breweries starting to move towards like even micros who poo-pooed it, just like in the wine industry, they kind of they didn't want to talk about getting away from corks and using screw closures because that was associated with crappy wine. There's been a huge shift in both those industries. You see craft beers moving to cans now, a properly filled, a CO2 displaced, fobbed and lidded can that's devoid of oxygen during the packaging process actually has a great shelf life and has come light years beyond where it used to be years ago. So some of the best beers are now actually in cans, just like some of the best wines you'll find with screw tops now, because they actually, it's a superior closure.
Yeah, I wasn't a fan of Corona before and definitely not now.
Hey, John,
Terrible.
is that your brewery in the background there?
Yes, that's actually in my basement. I have a 300 square foot nano brewery in my basement.
Wow, I'm jealous.
And I have a canning machine now.
Oh boy.
I think
Hey,
we had
John,
a question.
follow-on question to
from,
that.
Yeah, go ahead,
Yep,
Mike.
Go ahead, Mike.
Hey, hey, John. So kind of a follow-on to kind of the whole oxidation issue with hops is, have you ever grown your own hops for like finishing hops, and do you see it kind of worth the effort for doing that?
Well, so Chris can answer that question because Chris has Cascade growing in his backyard, but he keeps giving them to me because he doesn't brew enough. And yes, I do have hops grown in my backyard. So my hops that I have are wild. They're New York State hops that were made, I think, prior to the big mold or whatever it was that killed all the hops way back when. So New York State used to be the hub for hop production in the United States way back when. And then some disease came through and killed them all, and they moved it out west. And so now it's all, yeah, there you go, Terry. And so now it's all moved out to the West Coast.
State. Yeah.
Yeah. But so now, when I moved to my house, I was walking the property line out back, and looking around, I'm like, there's all these hop plants growing on here. It's all just escapees from that didn't get killed by the pests back in the day. So my hops, depending on what kind of beer you want, my hops are either perfect or completely not right. If you like a Pilsner, I got the hops for you. But if you like one of these American Pale Ales, like a Cascade hop or one of those flowery ones, or any of these new NIPAs my hop has no aroma and no intriguing taste, but it's got that bitterness and it's got that a little bit of the spiciness that the noble hops have. And since it's been growing here in the wild, that it loves these, and so they're just amazing. They grow during the hop growing season. It's like 3 inches a day. It's crazy.
Like weeds, yeah.
Yeah. I do have Cascade hops here, and they don't grow for crap in my backyard. Chris, yeah. I don't know. He does something special to his backyard to get them. Chris, what happened to you this year?
They don't grow well in the Bay Area for me, so I've always wound up having to buy them.
I find the trick is put a bag of cow shit on top of every one of your planting places every fall, and I've got half a dozen of them, and I have probably a 15-gallon tote next to me here. I'm putting the finishing touches on drying them. That's what I just held up. That's all Cascade I grew here, and it goes bananas when you hit it with cow dung.
Yeah, in terms of practicality, though.
Go ahead, Chris.
Oh, in terms of practicality, I mean, I get a lot of hops out of it, but you get I have 2 or 3 plants. You get 2 pounds dry. That's a good amount, but you could probably buy that for $40, and I spend a whole day picking little hop flowers off my plants. So
it's always that make-buy issue, isn't
it? Yeah.
But Mike, I should note the one thing you can do if you've got your own hop plants is make a fresh hopped beer. And you won't be able to make a fresh hopped beer if you don't grow your own. You can't, unless you've got some hop grower somewhere nearby. You can't make your own. The Sierra Nevada? What's the fresh hop Sierra Nevada? The winter beer. Their winter beer.
Oh yeah. What's
somebody here?
Species.
Come on. What's
Yeah,
the beer?
it is.
The test. Sierra Nevada's winter beer. The red label. Only comes out one time a year. No,
Celebration.
no. It only comes out one.
Celebration ale.
Celebration.
Yeah.
Yeah.
Celebration.
Celebration ale. Had to think about
So
that.
if you want to make
the red bottle.
Yeah, the red bottle. If you want to make your own celebration, you've got to have hop plants in your backyard because you pick them off the plant and you put them in your beer.
So, but what is that? Are you just reducing oxidation? Like, why is such a fresh hop beer so much different?
So you don't go through that whole drying process. So it has a different flavor profile. All the base chemicals are there, but you get it's just a fresher taste. And so for people who have not tasted Celebration, Sierra Nevada Celebration, it's coming out pretty soon. And if you see it at a store anytime in the next month, you should just get it and bring it home and drink it. It's the hops, and the hops are oxidized, and it goes off after a while. The Celebration from last year, even if it's been sitting in your refrigerator the whole year, isn't as good as it was when you bought it. But that's so fresh hop beers. You don't see it very often because the timing has to be perfect in order to harvest and then use it when you're brewing right then. Kind of tough.
Awesome.
So I've given talks, so I have actually up a presentation that I had given 25 years ago on how to brew. It's my yeast talk from 25 years ago. Everything has changed since then. Whereas 25 years ago, when I taught everybody how to make their own starters, that was a really big advance. Now the dry yeast packets have really come a long ways. And so you can, with a dry yeast packet, really introduce, but back in the day, we did counts on the packets, the little, the dry yeast packets. And you were maybe 1,000-fold under pitching when you were using those to start beers way back when. And so you were double, and then finally it gets to a Kreuzen where you can see bubbles coming up and so many off flavors. But these days, the yeast packets are really good. You can do smack packs, but the smack packs that people use, even if you get one of those big smack packs, it's not enough to really start out. Like I said, since you want to pitch your yeast in 2 doubles before maximum density, for a 5-gallon batch of beer, you'd need a 1-gallon starter. Right? That's like 20% of your beer is in a starter, is the starter flavor that you're adding in. So it's kind of tough to get the right number of yeast in to start your beer by making your starters yourself. What I'll do is I'll do a half-gallon or a gallon as a starter, but I'll let it go to full Kreuzen and then put it in the refrigerator overnight so that they all crash out, so I can dump the whole gallon off the top and just take that trub from the bottom and use that as my starter. But then it's really important that you oxygenate your wort before you dump those in so that they can kickstart.
And John, we refer to a smack pack. Is that the ones with little glass ampules inside of the foil packet?
So they definitely would not use a glass ampule inside of a foil packet, but it is a little sealed plastic bag. So yes, there's a little sealed plastic baggie with media in it that they throw in, and then they squirt in a yeast culture, and then they seal that up and they mail it to you. So when you get it, you lay it down and you try to figure out where that little baggie is, and you smack it to pop the bag, and now the media is mixed in with the yeast, and you give that a good shake and you let it sit. But there's like, I don't know, 35 milliliters in there, 50 milliliters in there, maybe 100 mils. The number of yeast that you're going to get. So I'll do a smack pack, but then after it, a day later when it's already gotten to high Kreuzen, I'll dump that into a half-gallon starter wort that I make myself. And I'll let that go to Kreuzen. And like I said, I'll put that in the refrigerator, crash the yeast out, dump the top off, and then take that slurry from the bottom and use that as my starter for my beer.
Okay.
But again, if you want to make a beer that's really going to impress your friends, or you're going to enter it in the competition, I'll then use that first beer as my starter beer, and that'll just be some beer that I'm just going to drink when I'm just drinking something for the heck of it. And then the next batch of beer, we'll use a quarter of that trub for the next one. So I'm going to say I'm going to brew on a Saturday, and then I'm going to brew again on a Saturday, and I'm going to use the trub from the first Saturday's batch as the starter for the second Saturday's batch, and that's going to be my really good beer.
So John, we've seen things like next-gen sequencing and some of these new kind of almost lab automation technologies emerge. Do you see any future in that in beer production or maybe homebrew in the future?
So yeast genetics is really complicated because all of these yeast strains have been grown for the flavor profile that they make. And, whether they attenuate or they don't attenuate, whether they flocculate or they don't, make sure they don't make diacetyls when they grow. You can buy the American ale yeast, the one that they use for Sierra Nevada Pale Ale. That's a really clean yeast. That doesn't make any flavors. There's the London Ale 3 yeast, which is the one that's for the NIPAs. That's the one which you brew at a slightly higher temperature, you slightly under pitch, and then during that course of it sweating, while it's going through a couple more doubles than you normally want to do, it makes a bunch of esters and polyphenolics. And those esters and the polyphenolics are those juicy bits that are in all these juicy beers that you hear. So the hazy juicies are all with special yeast strains that are grabbed because they sweat just the right chemicals that make your beer taste like they've had a tangerine thrown in them or something. Which for me is, it's kind of crazy to think about, but back in the day, I was all into steam beers, right? Steam beers for a while was big. Take your lager yeast and grow it at ale temperatures, and the sweat that it makes a beer that is your anchor steam beer. Now they got NIPAs, which is the same thing. You take an ale yeast, you make it sweat, and it makes esters, and they're all juicy. And when you mix them with all these newfangled hops, you get all these crazy juicy flavors, and then you've got that powdery mouthfeel from all the oats and wheat that they use in there. Well, I forgot your question.
No, I think it made it a little more complicated. So, I mean, it's more of a phenotype.
Yes.
Process, right?
So most of the brewing yeast, if they do genotyping on them, it turns out that they're polyploid. So they've got 17 chromosomes, but they've got like 4 copies of chromosome 3. And so it's really hard to do genetics on a yeast. It's not like us, where if we end up with, polytriploid, you end up with Down syndrome, right? With yeast, they don't have to think, so it's okay if they double up or quadruple up or whatever on different chromosomes. And so because they don't have single copies of each one of their chromosomes, it's really hard to do manipulation. And because making all of those right flavors is all of these pathways, going in and screwing up one pathway, now you've got all these different steps that comes down, and now you've messed up with some enzyme here. These things, the yeast want to figure out what to do with this, pyruvate. So what's it's going to do with the pyruvate if it can't break that down further? Well, it's going to shunt it off into some other salvage pathway, and you're going to end up making all these weird chemicals. And so genetically engineering yeast to do something like, so for instance, genetically engineering a yeast to not make alcohol, so you can make no alcohol beers. Really hard to do because you got to do something with those carbon chains. So that, I think that's the reason why it's so complicated to make new yeast strains without just doing batch after batch after batch, and trying to get the trub from the first that one and the next one and developing some kind of flavor profile. You probably could if you had a mass spec and you were doing it in small containers and you were looking for certain chemicals to get, but using like Cas9 CRISPR or something like that to do genetic engineering to change pathways, that I think would be is really complicated because of the polyploid nature.
Okay. So it's more like the game of selecting the right. We got a question here. Is it feasible to run a home brewing batch in a wine cellar? I'm in a wine growing region. That's a good question.
Well, sure, you can make beer anywhere you want. Just don't spit in your containers.
Is it the right temperature? I guess you'd have to adjust the temperature a little bit, right?
If you're home brewing, if you're comfortable in the room, yes, the optimal temperature is somewhere in the 60s. And well, I don't know, what temperature is your wine cellar? I mean, if it's like 39 degrees Fahrenheit, that's perfect for making your lagers. You just have to wait longer.
Yeah, that makes sense. So what you're saying is you should keep your beer in the room next to you is what you should do, right? Just in your room.
I think the question may be related to the fact that there's this yeast floating all around in the air there. And so you wouldn't want to do a natural fermentation Belgian in there because you're just going to get wine yeast that drop in. But when you put your starter in, you're putting in, trillions of yeast in as your starter. And so if, a couple hundred wine yeast float in while you're doing transfers, that's not going to do anything bad to your beer.
Okay.
And really, if you're making a 5-gallon batch of beer and I was going to say you're not drinking it fast enough if those yeast can have any long-term effect on it.
So this is a good question though. What is the kind of the longer-term impact of temperature? If you're in a place where you can't necessarily control temperature, or there's maybe the air conditioner comes on and off, is that going to affect the beer or the final alcohol production?
Are you talking about during the brewing process, during the fermentation process, or after it's been kegged or bottled?
Let's say during the fermentation process.
So certainly if the temperature is going up above 72 degrees Fahrenheit, up into the 80s or 90s, that will definitely make your yeast sweat. So you want to think about what kind of beer you're brewing. If you knew that you were going to end up with occasionally warming up and cooling down during it. So first of all, I'd make sure I had a really good starter to make sure your beer is just fermented as quickly as possible. And then I'd maybe use a strain like the American ale yeast, which is really clean. It makes it has hardly any flavor contribution at all to the final product. But I mean, the reality is your fermentation should really be about 90% complete in 24 hours if you've got a good starter in there, and then it's just finishing it off during the next 2 days.
And for
I think 96 hours is the standard turnaround in commercial breweries, I believe, with good starters.
96. So for someone who's like just getting into beer, home brewing, what type of beer do you think they should start off with?
You should brew extra light Coors Light. It's the hardest one ever to make. So really, through the years, I've had so many beer snobs tell me that Coors Light, Bud Light are just they're just ridiculous. I'm like, they might be ridiculous, but you can't make it.
That's right. There's no place to hide in a Pilsner, in a light Pilsner. There's no place to hide.
Yeah, a light lagered Pilsner, that's redundant, lagered Pilsner. But, so if you're lagering, the reason why you use lager yeast is because you're trying to make a beer with very little flavor. And the lager yeast eat some of the, I started out this by saying the starch gets turned into sugar and the sugar gets eaten by the yeast. But some of the starch doesn't get turned into maltose or glucose. It gets turned into maltotriose and maltobiose, these weird other sugars that yeast don't eat, that ale yeast don't eat. And these minor sugars have a little bit of a bite to them. And ale yeast don't eat those. And so those are all in your ale. But your ales are normally kind of hopped up a little bit. You don't really taste them all that much. Your lager yeast do eat those. And so now you end up with a much smoother final product. And now just a little bit of those noble spicy hops really kind of shine. And you end up with a nice clean beer with a nice clean finish with a little bit of a spicy hop. And so that's your Pilsner Urquell, you know, those nice Czechoslovakian Pilsner style beers. And what was the question again?
Is that the one that people should start off with, or should they start off?
Oh, no. So if you're going to start out, make a stout. Make a stout.
Okay.
So much crap in there. You'll never taste anything wrong that you do with your yeast. You won't taste any difference in the water hardness. It hides all of your mistakes. And so, you know, so if I was going to say, as a home brewer, as a starting home brewer, if I was going to define my first few batches, I'd make a stout, then I'd make a porter, then I'd make maybe a brown ale, and then I'd make maybe an IPA, crap loads of hops in there, all these things with really strong flavors. And then once you get really good at it, then you go make your Pilsner.
Okay. So the Pilsner
start with an ale.
Awesome.
Coleman, weren't you trying to brew a batch with some yeast you got off a tomato or something a couple years ago? What happened with that? Did it work out?
I've got 3 wild yeast strains, which are doing okay. I tried with an avocado, and that was a mistake. So do not try avocado wild yeast. But I've got a couple off of a grapefruit and one off of a jacaranda tree flower, which turned out pretty good. So I'm not sure, you know, I'm not as accomplished as most of you guys out there, but it works pretty well with my little, you know, Belgian style ales. And I think, John, correct me if I'm wrong, but the wild yeast tends to go well with that style of beer, right? The Belgian style, spicy, you know, a lot of fruity esters produced, those kinds of things.
Yeah. The Belgian, so the Belgian style definitely is, especially if you just let God take care of it. It's pretty funky, but it normally has some bacteria in there. Those Belgians, they've got yeast and bacteria that are dropping in, and so I don't know how you make your starter off of a fruit. I don't know if you're plating it out and picking yeast colonies, or you're just.
I cloned this one out using a really highly hopped wort, and so it kind of selected for did your starter pretty much. I threw an orange juice, or you know.
But you didn't plate anything out and go to colonies.
I did. I took out that's the flocculation layer, plated it out, and just kind of guessed at which colonies I should pick.
I see. So you were picking yeast. So you're plating things out. You're looking at them, and you're picking colonies that are yeast, and not like lactobacillus or something weird.
No, I talked to someone at Stone who kind of does their colony counting, and they said what to look out for, the nice fluffy morphology when you're doing your colony picks. And so I had a couple, you know, nice fluffy colonies. I punched those out and tried them out, and I think I just got lucky. So we'll see. But that's in my -80 here at the lab. So.
So it was funny when Juice Bomb first came out. You know, they say that it's unpasteurized and there's yeast in it, and that's part of what makes your their hazy beer hazy. So I took some and plated it out in the lab, and there were blatantly 2 different yeast strains in there. One made this sort of yellow creamy, sort of smaller colony. The other made these really bright, almost white, tall, fluffy colonies. And it was in very distinctly, it was in a 9-to-1 ratio. So if you count the colonies, there was 10 of these and 90 of those. And so I actually took a picture of the plate and I sent it to the Sloop Brewery. And I said, I don't know how you guys brew, but I just wanted to let you know I got, and I described to them what the beer was, where I had purchased it, and on what date. And I was like, and so there's no question that there are 2 different yeasts growing in this beer. And so it of course turns out that the one that was 90% is London Ale 3, and the one that's 10% is a wheat yeast, a wheat beer yeast.
Okay.
And that's they specifically do it in that ratio because it makes the right flavor profile when you brew it at 70, 72 degrees. It makes all those esters and all those fruity bits.
Okay. Have you brewed with multiple strains before? Like at the same time?
Never before a NIPA.
Okay.
Never. So I take it back. So I have lagered beers that I grew as an ale. So I would ferment with American ale yeast, which is a very clean yeast. And then when it's done, I would throw in a lager yeast and stick that in the refrigerator for a month to allow or not a month, less than a month. Because you know that the only thing the lager yeast are doing is eating those residual sugars that the ale yeast didn't. So why don't you just make a clean beer and then lager it off with your lager yeast? So that's the one time that I have mixed the 2 together. It was too much work to really validate doing it in the long run.
There we go.
Yeah, I think it's just easier to start off your lager yeast at a slightly higher temperature, get them going really well, and then you're still going to end up sitting around for almost a month once you put it in the cold room. So,
hey John, have you ever had the pleasure of running into diastaticus?
No, I have not.
That's the one that's normally responsible for bottle bombs in home brewers and in commercial breweries, where in packaged products everything seems to be fine for a short time, and then slowly those cans will swell or the bottles will build pressure, and then they'll just start detonating randomly. And that's usually because you've got a wild diastaticus infection, and that one, it doesn't stop at the dextrin line. It keeps chewing very slowly on the dextrins, and it keeps moving its way up. So that's what causes a lot of beers to blow up. And when you get it, it's hard. You really have to sanitize like crazy to get rid of it.
I think that's what shut down the Pyramid Brewery here in the Bay Area years ago.
Yeah, when it happens, I mean, there's very few. There are a few styles where it's actually the yeast that you would select, but it is almost nobody would do it on purpose. And in a commercial brewery, you let that genie out of the bottle, and you're going to be chasing it around forever trying to sanitize your way out of it. It's they could just ruin a commercial brewery for a period of time if it gets loose.
Man, if that happens, you're going to do this, like, so at the very start of this, I was telling people that my local bar closed down because multiple servers came down with COVID, and the local, the fast response team. So the local fast response team, the people that clean up when your water main breaks and your basement floods and stuff like that, they've completely changed their whole business model to coming in and bombing people's houses that are businesses that have employees that show up with SARS-CoV-2. I could imagine that a brewery would want fast response to show up and just bomb the whole place, kill every single surface and every container. And I mean, what do you do if you've got $1 billion worth of metal that has had that introduced into it? You got to sterilize
and it
everything.
doesn't matter the amount of polish you have on your stainless either. It's like there's always micro cracks and little scrapes and things and the nooks and crannies for it to hide in. So the best thing is heat. If you can get it to everything, a good hot wash will do miracles in a commercial brewery. But there's all sorts of chemistries that will do damage, but you have to have a little bit of a surfactant and wetting agent in there to get it into those nooks and crannies. Cause it's, it sounds weird, but you have to make water wetter for it to get in and do its job, because it can't get to everything. So heat is best if you can do it. There's a lot of chemistries that'll do it, but it's if you get bored, look up, I know you're a genetics guy. So the STA1 gene, that's the one that's responsible in the beer in the beer line of yeast. If the STA1 gene is present, that's the one that encodes for my understanding. This is my layman's understanding of it. That's the one that'll turn a yeast into a dextrin loving yeast, where it'll keep going up the like usually there's a shelf. It hits the dextrin line and it stops. That's as far as most commercial yeast will go. But some of the wild variants will keep on chewing, and they'll do it slowly. And so you end up building more CO2 in the container after it's been packaged. And that's how you end up with these commercial nightmares with bottle bombs and blown cans and bad shipments that kind of creep on you over time after shipping.
The other thing that can create problems, and I've got a publication on this, is that there is a low level of amylases in hop leaves, in hop flowers. And so when people are making these NIPAs where they're dry hopping, trying to pump up the aroma, and they're then extracting out some live enzymes, and then those live enzymes goes into the cans, and then the cans get sold, and you don't, or either the distributor doesn't refrigerate them, or you buy them and you don't refrigerate them. Those amylases become active, and then they start breaking down the residual starches that are in there, and you end up with exploding bottles. Well, everyone puts it in 60-ounce cans, so you end up with your cans exploding.
Yep,
so that was a little-known fact, was that there are definitely amylases in the hop flowers. And if you're doing a 5-gallon batch of beer and you're putting 8 to 16 pounds of yeast of hop flowers into your dry hop, just because you're crazy and you really want that aroma to blow you away, you have to be really careful that you refrigerate it. And that's why my local Save More Beverage place, the owner he asked me, he's like, why do these guys require that I refrigerate their beers? Why can't I just put it out on the shelf with all the rest of the beers? I'm like, because there's an enzyme in there, and it's going to turn those beers into little bombs. And so you need to refrigerate it so that the enzyme doesn't work.
Yep.
That's your own risk. Hey guys, we're at time, and I think this has honestly been fantastic. John, thank you so much. I'm going to put my email out on the chat. And so if you're interested in doing another one of these, I think this has been really fun. You know, there's a growing community of home brewers. So shoot me an email if you're interested, and we can kind of do a call too, because I know we've just kind of tapped into John and Terry and some of the other members' resources and knowledge here. But with that, I kind of want to close us out and just really say thank you. This has been really fun. John, thank you so much for your time. And if you're open to it, maybe we can do another happy hour.
Sure.
All right. All right,
I'm
guys.
always willing to drink beer and talk about beer.
Thanks, John. It's great seeing you again.
Thank you, Coleman. Thank you, John.
Thanks.
Thank you, everybody.
All
This
right.
was really great.
Yeah, have a great night, all.
Thanks, all. Take care.
Thanks.
Cheers.